Good morning. I want to thank Marsha for the opportunity to speak to you. Today, I'll be talking a little bit about my work, and my presentation is entitled Using Genome-Resolved Metagenomics to Understand Microbial Contributions to Ecosystems Functions. But before I get started, I'll first give a brief overview and introduction to my group. So I work in the microbiome research group in South Africa at the University of Pretoria. Our group currently has about 20 different researchers working at PhD and MSc levels. We have a broad range of interest, but before I talk about those, I'll give first just a broad overview of the microbiome and give you a little bit of a taste about what drives our interest in our work. So what exactly is microbiome research? Perhaps before getting ahead of myself, I should start first by defining the word microbiome. The word microbiome defines, or is the combination which defines the genetic material of an organism in a particular environment. So this includes microorganisms that may be linked to particular host, soils, water, both aquatic and marine environments, or even the air. So we know that microbes are essentially quite ubiquitous and are highly abundant. In any environment, there are at least 1 billion different types of bacteria. So these are present on your skin, they're present inside your body, they're found on leaves, they're found in the air, they're found in deep sea hydrothermal vents and other extreme environments. So most microbes are essentially very good, but we know disproportionately very little about these microbes. Instead, most of the research has focused on the microbes that have a potential to carry out or give us essentially bad ecosystem services. So why do we care about the microbes that, for instance, may be in the environment? You know that you inhale more than 10,000 bacterial cells per hour. So not even pollen, fungi, but some of these microbes are microbes that we essentially know very little about what their ecosystem services are. We know of those microbes that lead to allergic reactions. So for instance, it has been estimated that by estimates of around 10 million people in the United States have different types of asthma. And these, of course, lead to bad health outcomes. We do know that a very high diversity of microbes are present in any kind of environment that we look at. So the question is, why is there such a high diversity of microbes? What type of ecosystem services do these microbes provide, if indeed they do provide any kind of ecosystem services? Are these microbes essentially there, not conveying any kind of service? This is something that's probably unlikely. We know that these microbes are likely interacting with each other, and these interactions also foster the type of outcomes that we receive from these microbes. For instance, we know that the combination of microbes that may interact with particular plants promote plant growth promoting organisms, and these convey very beneficial ecosystem services in terms of their role promoting nitrogen fixation, for instance, in plants. So microbes play essential roles in natural environments. This very colorful tree that maybe someone would have shown in the workshop conveys what we actually know about the microbial tree of life. We know that shown in blue is where we are in the tree of life, and that plants are somewhere very close to us. So the interesting thing is that most of these very colorful colors and very colorful clades are essentially microbes that lack genomic representatives. And we know that these microbes exist because of genome-resolved metagenomics or metagenomics analysis, which rely on us being able to study microbes without the need to, without the requirement of bringing them successfully into culture. So from these studies, we've been able to gain a lot of insights. For instance, studies on microbes that are found in extreme Antarctic environments have revealed that even in such cold environments, that there are adaptations which facilitate microbial communities and their development in these climatically extreme environments. Our work in desert systems, for instance, has revealed very interesting insights regarding the ability of microorganisms to carry out functional services. So we know, for instance, that in extreme namib soils, that cyanobacteria essentially drive community composition and functional processes, and that these microbes are essentially the base of the food web structure in these very oligotrophic environments. Other studies in Antarctic soils have revealed very interesting insights. For instance, we've discovered that in essentially pristine Antarctic soils, there's a huge diversity of historical antibiotic resistance genes that are found within soils that have very little contact with other humans. So these are functions that are found in these soils, in these ancient soils, where microbes convey essentially very interesting mechanistic interactions. And our work really aims to understand essentially what the role of these microbes are in terms of driving biogeochemical cycling. And we're also interested in understanding what the potential impact of these microbes may be to climate change. There's been very interesting studies that have been done in marine environments that have shown that, for instance, microbial ecosystem services can present additional feedback mechanisms that may exacerbate the effects of climate change. So with this slide, I essentially want to show you that microbes are very important, irrespective of the environment that you look at them from, and that we know that they play essentially important roles. For instance, I've talked about the role of bacteria, but we know that fungi, for instance, also play important roles in driving decomposition of organic matter. So irrespective of the environment, we know that microbes play a very leading and dominant role in driving the ecosystem services. So our research has got about 4 different thematic areas. We are interested in research that aims to essentially carry out a bit of bioprospecting, where we search and characterize new enzymes from various terrestrial and marine environments. We've got research that focuses on understanding the ecology of extreme and local environments. So these local environments may include places such as host-associated microbiomes in South Africa. A lot of our work focuses on characterizing the microbial processes that underlie the evolutionary adaptations of microbes. And we aim not only to understand these evolutionary adaptations, but essentially to also see how we can use the natural adaptations of microbes to be able to tap into the biotechnological importance, biotechnological traits that these microbes may be able to provide. The third avenue of research is that we focus on microbiomes in what are called geographically strategic marine environments. So these are places such as the Atlantic, the Indian, and the Southern Ocean. We've also got research that focuses on understanding human gut microbiomes. So we ask questions, for instance, that are linked to understanding how people in rural environments may have gut microbiomes that are specifically adapted to their particular diets or geographic locality. So the current projects in our group are the following 5. We've got a project that's looking at the microbial ecology of South African soils. So this includes everything from isolating potentially novel bacteria and fungi from these soils. We've got projects that look at human gut microbiomes for urban and rural individuals, as I've mentioned previously. And we've got a project which aims to understand or provide an understanding of the ecology, evolution, and function of marine microbiomes. And this project focuses quite a lot on the Southern Ocean and the South Atlantic Ocean. We've got projects that aim to elucidate microbiomes in the South Atlantic. And this is a project that's currently being carried out with partners in the EU and also in Brazil as part of a Horizon 2020 funded project. And then the last project that we have in our group is a project which aims to elucidate the microbial ecology of Antarctic permafrost environment. So we know that as a result of climate change, there is a drastic destabilization of what were essentially very stable systems, with permafrost remaining unchanged for many years. But we, as part of the warming that is occurring in the Antarctic, we expect that this may lead to essentially the exposure of microbial communities, so both bacteria, viruses, and fungi that may have been dormant in these soils. So we've got a series of projects where we've essentially taken permafrost from Antarctic soils and have simulated warming in the lab, and we're trying to understand how the diversity of these microbes may change, understand important questions related to host virus interactions within these systems, and also try to elucidate the functional dynamics within these communities. So what you can see from the last slide is that our research focuses quite a bit on polar and marine environments. So there's a strong emphasis on polar marine environments because, as I mentioned a little bit earlier, that we have a very strong geographic advantage as South Africa. South Africa has about 3,000 kilometers of coastline, and we're surrounded by the Indian and Atlantic Ocean, and also to directly below us, the Southern Ocean. So we have started several interesting studies that have focused on the molecular ecology of these environments. And again, these studies also look at understanding the potential biotechnological importance of these environments. So if you can see from here, that's where we are, and you guys are all the way over there. And South Africa, as you can see, is surrounded by all these environments. We actually have a base in the Antarctic, and we've maintained some kind of presence in Antarctica since essentially the late 1950s. There's also a very strong scientific justification for these studies. So we know that the oceans are important in transporting nutrients, heat, and because of this role, they essentially regulate the climate. The oceans are also responsible, and particularly the Southern Ocean, for sequestering the majority of the world's carbon dioxide. So they do this essentially through two different mechanisms. The first is the solubility pump, where a cold carbon dioxide containing water essentially sinks to the bottom of the ocean. And then the second is what is termed the biological pump, where inorganic carbon dioxide is converted and eventually sinks to the bottom of the ocean. So we know from previous studies what the role of phytoplankton and zooplankton may be in terms of regulating these processes. But we essentially know very little regarding the role of bacteria, archaea, and fungi in converting inorganic carbon dioxide and essentially contributing to the biological carbon pump. So our studies aim to essentially go from a broad spectrum of characterizing phylogenetic diversity that's linked to microbes, and essentially using a series of either in situ experiments or lab-based experiments to ask more specific questions related to their functional attributes. So the oceans are important, as we all know, in terms of their role in sequestering heat, and they are, key areas in terms of buffering the Earth regarding the effects of climate change. So we know as well that the Southern Ocean is important, not only as a provider of goods and services, but also in terms of its role in sequestering most of the world's carbon. I think estimates suggest that about 40% of CO2 may be sequestered in the Southern Ocean. So the last 2 points I'll mention very briefly, that South Africa has got a strong push in terms of driving fundamental research in Antarctic systems. We've contributed towards the Scientific Committee for Antarctic Research since at least the late 50s, and we play a very important regulatory role as part of all of these scientific bodies. And part of that role is to carry out Antarctic research, which we believe not only benefits the country, but also benefits the world in total. And we've also been, in our group, involved in some efforts to essentially contribute towards what would be an international consortium looking at the role of the microbiome, but also linked to this, exploring the biotechnological links from the ocean so that we can sustainably begin to harvest the products that we derive from the ocean. So essentially, in our group, we are able to carry out all of these projects by using a combination of bioinformatics and also by using computational analysis that we access through high-performance computing resources. So for most of our projects, we start from a point where we collect samples, and this is typically followed by DNA extractions and some form of sequencing, either amplicon-based analysis with 16S sequencing, but in this example that I've shown here is that we'll typically also do some shotgun sequencing. Part of this would also then include doing some computational analysis, whether it's through assembling the context that we've generated, and then this would ultimately go into different questions where we essentially try and look at the diversity within these systems. So part of the diversity studies now would allow us to reconstruct bacterial genomes from our metagenomic data sets, so through genome-resolved metagenomics. And then part of that would include also some functional annotations where we look at either a combination of functional genes, and in some studies we may also then look, for instance, at particular antibiotic resistance genes, and this would be done through specific databases such as RGI, and would also then carry out functional annotation using internationally available platforms such as DrumV. The other part of our studies, this is an example that I'll mention now, is that a lot of these computational analysis either allow us to directly carry out the generation of metagenome assembled genomes. And for this, we've been using programs like Atlas, which are available. And these allow us to more or less automate and reproduce some of the pipelines that we follow. And I'll, in the next slide, just give you an example of one study where we've used a combination of the tools that I've shown here to look at the phylogenetic diversity and also look at characterizing, for instance, specific viruses, so crassphages that may be linked to particular hosts. So this, again, is an overview of some of the workflows that we use for our metagenomic analysis. So the first study that I'll talk about is a study that has been done this year by one of our honor students. So in this study, we wanted to look at the microbiome of South African rivers. So we know that, over the past 20 plus years, even longer, that fecal coliform counts have been the standard for determining the quality of water. So in this study, what we wanted to do is essentially use a different molecular marker, and we identified the crassphage viral sequence as that particular marker, to assess how dominant, how prevalent it might be in South African aquatic environments. So what we did is we sampled about 12 different sites, or 6 different sites, where we looked at pristine and waters that were found on top of a mountain, and then we surveyed this water as it went through, on one end, agricultural waters, and then on the other end, a village without adequate sanitation. So what we did in this study is we used metagenomic analysis and the workflows that I described in the last slide to look at the phylogenetic composition of microbes that are present in this soil, water sample. And what we found is that we found a diverse consortia of bacteria and archaea that were present within these waters. We essentially found also a high proportion of crassphage sequences. So what was interesting is that in water which was estimated or determined to be free of coliforms, you can essentially see markers that are specific to crassphage viruses, which are known to be linked to gut microbiomes, and so indicate in some way that the water may be inadequately treated and not really safe for human consumption. So what we found in this water is that there were diverse consortia of crassphage viruses that were present. We found essentially novel clades that were not similar to anything that was previously characterized or present in sequence databases. So from this one study, we obtained over 200 potentially novel bacterial genomes, and these were high quality and also a combination of medium quality metagenome assembled genomes that we are currently characterizing further, and these will then be used to map the specific viruses to particular host, to allow us to get some kind of idea in terms of the evolution of particular prokaryotes within these systems. So in another study, we also were interested in understanding the taxonomy and functional diversity of marine waters. In a paper that you can actually find on bioRxiv, which is currently under review, we explored the euphotic zones in the South Atlantic Ocean. So in this paper, what is most interesting is, in addition to the very high diversity of potentially novel bacteria and archaeal taxa, what we found is that some of the mechanisms linked to phosphate acquisition were essentially driven by viruses. So viruses played, through the actions of auxiliary metabolic genes, play a strong role in influencing biogeochemical cycles that occur in marine environments. So if you can, I'd encourage you to have a look at this paper if you want to know much more details. In the last 5 minutes of my talk, I'll talk a little bit, give you some insights also how we've used genome-resolved metagenomics to look at microbes that are in the Southern Ocean. In this work, which is also under review, carried out by PhD students, we looked at microbes that are found in the transition between winter and spring, and specifically try to understand the phylogenetic composition of these microbes that are linked to a spring bloom. So what we found is that by surveying an area of over 2,800 kilometers in the Atlantic center of the Southern Ocean, we were able to provide what is, we believe, to be the most extensive gene catalog of Southern Ocean microbes. We found over 300 high-quality genomes that were potentially novel in the South Atlantic Ocean, in the Southern Ocean, and these were genomes that were previously uncharacterized. I haven't shown it in this slide, but we've got over 10 million genes that are essentially unique to the Southern Ocean, and these were compared to a gene catalog from the Global Terra Oceans survey, and also to the Malaspina survey, which were previously published. And this shows a very high degree of endemic bacteria which are present in the Southern Ocean. So I won't talk about this in much more detail, but the paper is available if you are interested. So we've also got a series of projects where I spoke a little bit about the biodiversity surveys that we've done, but now we're trying to use machine learning approaches to essentially do some bioprospecting of these novel bacterial and archaeal genomes that we found in our data sets. So for this, we're using a combination of machine learning approaches that where we are either establishing new machine learning algorithms or using existing ones to carry out some kind of bioprospecting analysis, where we're doing surveys of potentially novel gene targets. So these gene targets, once they're identified, are then, depending on the length of the gene, synthesized and functionally expressed and characterized in the lab. And a lot of this work, is revealing enormous biodiversity, and it shows that we essentially have an untapped biological resource that's present within the oceans. And this could be, the future basis for launching startup companies. And this is part of the work that we're exploring at the moment. I don't have time to talk about this, but at the end, I'll just thank you all for listening, and thank the funders that have facilitated this work. And I am available. I should have put up my email, but I am available in case any of you have any questions. Thank you for listening.